Hello, everybody. Welcome to session number 2. I'm glad to have you all here. I just want to go ahead and introduce Dr. John March, who's going to be presenting Neurosciences 102. And he's our new faculty leader here in Neurosciences Medicine. He's a professor of psychiatry, behavioral sciences, psychology, and neuroscience at Duke University Medical Center. He's also the former chief of division of child and adolescent psychiatry at Duke, and he's the principal investigator for the CAPTAIN study here at the DCRI. He is widely published in the areas of OCD, PTSD, anxiety, depression, ADHD, and pediatric psychopharmacology. And an interesting fact, before training in child and adolescent psychiatry, Dr. March used to be a family practitioner in rural Montana. So let me give the floor up to Dr. March.
So thank you, and the one thing that Maureen didn't mention is that I'm married to Margaret, who is sitting over in the back there. And if I say anything stupid, because I have all these letters at the end of my name, she says that the actual thing that ought to be back there is KBSA, which stands for King of the Bullshit Artists. So what you hear, you should take with a grain of salt, or ask Margaret about it. She'll tell you whether it's true or not. So what we're going to talk about today is something that even as recently as 2 or 3 years ago I would never have had the temerity to bring up, and that is the possibility of a world without mental illness. If you go to the National Cancer Institute website and look around, you'll see that the motto is a world without cancer, that the mission statement for the Cancer Institute is a world without cancer. And nobody in their right mind would have thought, even as recently as 2 or 3 years ago, that we could begin to think about a world without mental illness. That is, the scientific challenges were so immense and so daunting, and the lack of knowledge so apparent, that even begin to think about prevention and cure seemed off in the very, very distant past. And now everything has changed. And there are a couple of things which have produced this change. One is dramatic advances in the understanding of the molecular neuroscience underlying mental illness. And the second is an understanding that these phenomena are developmental. That is, mental illness is something that happens first in kids and then in adults. And this ability to think along trajectories from neonates all the way up to geriatric psychiatry, and to do it in a way that connects genes to cells and cells to systems of neurons and systems to people inhabiting the context in which we all live, is really a brand new idea, and it's what allows us to think about a world without mental illness. So what I'm going to do today is to talk to you kind of about this, and what I want you to do is not to think about the details of it, because the picture I'm going to present is incomplete and I'm sure contains some things that are either inaccurate or untrue just because of where we are knowledge-wise, but rather just focus on this kind of big idea of this grand vista. It's like we've been given in neurosciences medicine over the past couple of years, we've been given the equivalent of a Hubble telescope. So what you want to do when you hear this story unfold is simply to think about what it would look like if you were looking through the Hubble at the brain of a mentally ill patient. Now, these are my disclosures. I'm not actually going to be talking about psychopharmacology in this talk, but I have lots of relationships with different people. And the important bit of information on this slide is that for the last dozen years, I have not participated in any speakers bureaus, so I don't do any promotional work at all with industry, and wish my colleagues actually would adopt the same position. So we'll talk today about what is developmental neuroscience and why it matters. We'll talk a little bit about the perspective of developmental neuroscience as we move from genes to cells, systems neuroscience, and people in their context in which they live. And we'll talk about the implications for all of this for how treatment will be organized when we hit 2020 or 2030, when the whole landscape of medicine is going to look dramatically different from what we have now. So everybody knows that development matters. This is the anatomy of a toddler's brain. There's a big, prominent line center up here. I'm not sure this is limited to toddlers. The whole world is whining. So this is an adolescent brain. Things happen across development, and you probably have a hard time seeing that, but this is pin code, two roads diverged in a wood. That guy was cute in ballet. First kiss, I don't know, a bunch of different things in here. But things happen when you cross this boundary from middle childhood into adolescence that dramatically reshape the way the individual experiences him or herself, and also sets up a vulnerability window for mental illnesses like schizophrenia and social phobia, for example. So we know from the epidemiologic literature that if you were mentally ill as an adult, about 50% of the time you were mentally ill before you turned 14, and about 75% of the time you were mentally ill before you turned somewhere between 22 and 24. So the take-home message is that mental illness is a disorder of children, and once you get it as a child, unless you're treated with the best of evidence-based care, you're sentenced to be mentally ill as an adult and essentially have a chronic, a life of chronic waxing and waning mental illness on up into the geriatric years. This is, oops, I'm sorry, I did what I wasn't supposed to do. Just do that, yeah, oops. Okay, so on the left is the data on which that statement that mental illness is a disorder of childhood extending on into adulthood is based. It's data from Ron Kessler. And on the right is data from Phil Wong, who's the head of the Division of Services and Intervention Research at the NIMH, showing that from the time of diagnosis, which is well past the point at which the disorder's onset, 10 years from the point of diagnosis is the entry point into the universe of treatment. So we know that mental illness starts early. We know that if you don't get to it early, you're likely to have chronic waxing and waning mental illness. And even with that knowledge, most patients have a long time from the onset of symptoms to first diagnosis, and even longer to first treatment. Please.
What percentage of the population in their youth would be considered mentally ill?
At any given time among kids, if you exclude the simple phobias like fear of heights and snakes in the dark, it's 15 or 16%. Over a 6-month window, we'll have diagnosable mental illness. If you impose a severity threshold on this so that you want the kids who are unquestionably in need of treatment, then the number falls down around 10%. So that's 1 in 10 kids has something that any reasonable person would agree ought to be treated. And it's no big mystery why this should be the case. The brain is an extraordinarily complicated organ. Humans are relatively recent creatures in evolutionary terms. Modern humans are between 50 and 150,000 years old in evolutionary terms. The brain, in constructing itself, and we'll talk about brain architecture, hasn't had very much chance to adapt to the fact that we have all this capacity for language. And so there's lots and lots of parts of us that in a sense aren't in an adaptive way evolved in an evolutionary context. So it's no big surprise, at least to me, why there's so much mental illness. Now, translational developmental neuroscience, actually if we go back here, which is the context in which this talk unfolds, namely a advisory group of the NIMH Council that Pat Levitt, who's a developmental neuroscientist at Vanderbilt, and I co-chaired last year, is essentially calling for the development of a new field in which this developmental neuroscientific perspective now becomes the framework for all of psychiatry, not just child psychiatry, but child on into adult psychiatry. And we call that field translational developmental neuroscience, and it focuses on the trajectories of development as they're expressed across levels of analysis. That's genes. Genes run cells. Cells assemble into systems of neurons, or what are called hierarchically distributed neural networks. These systems of neurons then comprise the brain that we know that directs the emotional and cognitive and motivational and motor experiences that we have as individuals living in the context in which we live, in relationships, and so on. And then translational developmental neuroscience also, as necessary, moves across different experimental platforms. So there are some things that you have to do in a real living human being. If you want to know something about children, you can't study a rat. There's no rat model or mouse model of autism. There are models in the rat that are relevant to autism, but there isn't any such thing as an autistic or a schizophrenic mice. So what we need to do, or what we are in the process of doing, is understanding how to move across these levels of analysis and then be able to make inferences about what's going on at a molecular and cellular level, for example, because we can study phenomenon in flies, worms, fish, rodents, non-human primates that are relevant to human disease. And it's this capacity, this operational capacity, that has basically allowed us to think about a world without mental illness. Now, to accomplish this task is going to take between 30 and 50 years, and it is going to be an extraordinarily complicated and difficult undertaking. The human brain has 100 billion neurons, 100 trillion synaptic connections, and 3 million miles of axons just in your brain. About 30 million of these neurons light up every time that you see the figure of a face. That's an enormous population of neurons that are devoted to face processing. There are over 1,000 disorders of the brain and the nervous system, and we probably know about 0.1% of what we actually need to know to prevent and where necessary treat mental illness. But we have the tools to get there. Peggy?
Tell us what a neuron.
So I'll get there. I'll get there. So this is what we're going to be doing. We're going to start off talking about development. What do I mean by development? How do you build a brain? And then we'll talk about genes and why genes unlock the mysteries of cells and how cells assemble to form systems of neurons and talk with each other and process information, and how those processes, those information processes, are dramatically impacted by what happens in your environment. And this interaction between the environment and the brain as you build your brain growing up then takes us into the realm of clinical neuroscience, which is where we all work day to day at the DCRI. Now, the brain, as it grows, assembles from a single cell, an egg, into this very complicated architecture that connects the frontal lobes, where higher order information processing happens, subcortical structures like the basal ganglia, which is where habit learning and a lot of relaying around happens, the amygdala and the hippocampus, which are, this is terrible, which are down here in the anterior temporal lobe, which is where memory in the hippocampus and fear and anger processing, emotion processing happens, and out here in the parietal cortex, in the back of the brain here in the occipital cortex, which is where a lot of sensory information processes happen. So if you look at this process of normal development in the brain, Karen, if you can click that word normal, it'll pull up a movie. So this is the process by which the brain begins to form itself in terms of the prefrontal cortex. So if you watch what's going on, you can watch this wave moving from back to front. This is a normal brain. This is data from Jay Geed and Judy Rappaport at the NIMH. This is the normal process of developing from a 6-year-old into a teenager. And you can watch, it goes from the older parts of the brain, sensory processing, to the more complicated parts of the brain, back to front. Blue back here, and then eventually blue up here. So you can close that down. Now, the reason that it's important to know this is because this is the reference then. You think about these developmental trajectories, things changing across time. We need a normal reference to know when people start to fall off those developmental trajectories in particular domains. And when that happens, we label it as mental illness. Next slide. Oh, actually, I can do next slide. Here we go. So back to Peggy's question. You start out in the brain with a fertilized, not in the brain, you start out with a fertilized egg, right? And it implants and it begins to do magical things. And after a while, it becomes a blastocyst, and within that blastocyst, there are cells called ectodermal cells, which are out here, and they begin to look like neurons and move around to form the spinal cord, and the cells that will eventually become the brain, those two things comprising the central nervous system. To do that, they have to move around, so they migrate, they go to places they're supposed to go, and when they get there, they aggregate with each other, and they form different sorts of neurons, like the glial cells or neuron. There are actually over 100 different kinds of cells in the central nervous system, sort of major classes of cells. And having done that, they set up conversations with each other. That's called synaptogenesis. So an axon from one neuron will hook up with a dendrite from another neuron, and that synapse then becomes the on and off switch by which cells communicate with each other. And having gotten there, then a whole bunch of the cells that aren't relevant die off. That's called pruning. The process is labeled apoptosis. And then there's this final bit of rearrangement, and eventually you've got a whole set of neurons that are hooked up to each other where they're supposed to be, having very elaborate conversations. Remember, 30 billion neurons involved simply in face processing. And these processes then are what define brain architecture. Now you can see this if you look at embryonic or fetal development. This is the actual size of a fetal brain at 25 days. And it grows and grows and grows and grows. And out at 100 days, it's about this size. And of course, this is not the real size of a baby's brain. But if you look at this, the brain becomes more and more folded and complicated. It's actually quite remarkable how efficiently space is used inside the skull until you get this brain of an infant about to be born. Now, if you look at this same process over a longer period of time, this is now a schematic of the development of skills linked to brain function. You can see here at birth, babies can see and hear, so the visual and auditory cortices are actually quite mature. Out here over the first year of life, you begin to develop the capacity for receptive and then expressive language. And higher cortical functions, the ability to reason, to think, to make good decisions, executive function, don't mature. Here it looks at these as somewhere in the early teenage years, but in fact, we know now from more recent research that the brain of an adolescent, a 16-year-old, will not mature with respect to the ability to make good decisions until somewhere into the mid-20s. Now, experience has a big impact on this. It's not deterministic. In fact, the brain requires experience from about the second trimester on. The brain actually requires experience to successfully build brain architecture. So the things that happen to you were tightly coupled, completely coupled, absolutely coupled to our internal and external environment. And it's that coupling process that allows this brain architecture to unfold. So if you don't have things that impact the visual system, the eye doesn't have an object to relate to, then the visual system doesn't develop normally. And that's true for everything, including higher cortical functions that are involved in emotion, empathy, the ability to discriminate a happy from a sad face. All of these things require actual experience in order to develop normally. And here's a really good example of how you can screw this process up. This is the density of dendritic spines, which, remember, those are the part of the on-off switch at the synapse, as a function of having cocaine in the nucleus accumbens, which is the part of the brain that handles things that are rewarding. This is the addiction nucleus in the brain. This is a synapse, dendritic spines in a neuron that did not get cocaine. This is in a rat model. And this is the dendritic spine or branch process in a neuron that was exposed to cocaine. And you can see how many more dendritic spines are situated on this neuron that got blasted with a dopamine antagonist, a reward-based antagonist. And you can begin, just looking at that picture, understand why cocaine induces craving, because there's a lot more neuron out there looking for the equivalent of cocaine. Now, this is an example of how brain architecture, in fact, does change during adolescence. This is looking here in this slide, in this panel, at areas of the brain where volumes are smaller in adolescents than young adults. So you go to the back of the brain and everything looks pretty good, but you get out here in the emotional area of the brain and up here in the prefrontal cortex, which is where executive function rests, the ability to delay responding. You know, for example, when somebody makes you mad, the ability to say, well, he's just having a bad day instead of punching him. These things and the communication between these emotion regulation centers and the higher cortical centers, these things mature late in adolescence. And so the idea that, oh, he'll just grow out of it actually has some bearing in fact. And you can see the same thing, although I won't go into it. If you look at the sort of paucity of synaptogenesis, the amount or development or sophistication of the abilities of neurons to talk to each other, you can see in this rat model that they get much more sophisticated as you reach the adult brain as compared to the adolescent or less mature brain. So to summarize all of this, we now, instead of thinking about a 6-year-old, we think about a 5-year-old in the process of becoming a 6-year-old on the way to being a 7-year-old. We're looking at trajectories across time, not cross-sections within time. And within that, the brain grows and develops. And as it grows and develops, it acquires the capacity to see things, hear things, sensory information processing, to assemble that information so that it's biased as pleasant or unpleasant. There's an emotional reactivity component to it, to integrate that with memories of previous experience, and then to grow in skill with respect to making decisions about how we use that information to manage our lives. And when something happens, when there's a perturbation, either genetic or experiential, an individual who's vulnerable will then fall off this normal developmental trajectory, the green line, and that can happen right away, or it can be delayed. But eventually, when a patient falls off the developmental trajectory, everybody begins to notice. And mom starts to worry about the boy who won't sit still in class and isn't learning to read. The earlier you fall off, the bigger the gap, the more worrisome it typically is. So if you look like ADHD at age 3, it's much more likely that you've got bad illness than if it's diagnosed at age 9. And we think about then trying to return this child onto a normal developmental trajectory so that that brain architecture will resume growing normally toward adulthood at that point. Now, a couple of things about this. One is that these processes are fundamentally genetic. So virtually all, that's probably not too close to being true. It may be an overstatement. But they're, as far as we know, in all mental illnesses at this point, there are genetic vulnerabilities. And these vulnerabilities require environmental input in order to be expressed. So neuroregulin 1, which I'll talk to you about in a moment, has a dozen different forms. And one of those forms is expressed in the brain during fetal development and fetal development only. And it's a variation in that particular NR1 gene, neuroregulin 1 gene, in that fetal isoform that is the risk factor for schizophrenia. So it happens down here. Schizophrenia happens up here. Same thing is true for Alzheimer's disease. Teenagers who carry the ApoE4 risk allele for Alzheimer's, the Alzheimer's is very strongly genetic. And ApoE4 explains about 25% of the risk for Alzheimer's disease. Teenagers who carry this risk allele for Alzheimer's disease already show prefrontal cortical thinning, gray matter thinning, even though Alzheimer's itself won't show up until they're in their 50s and 60s. So again, it's a developmental process that begins very, very, very early in life. And if we have any hope at all in making a dent in it, we're going to have to reach downward in the age range. So go ahead and click that ADHD. So you can see this process of development and how it the developmental trajectory in these youngsters with ADHD. So again, out of Jake Geed and Judy Rappaport's group, this is a normal kid on the right, and this is an ADHD kid on the left. And when you look at blue, what that means is prefrontal cortical or cortical gray matter development. So if you watch this, what you see here is that this stays light blue about 3 years longer than this. So to get out here into this adult prefrontal cortex, capable of making good choices, not being overwhelmed with novelty or super rewarding stimuli or getting really mad and responding impulsively, to get to the same place that you would be in a youngster without ADHD takes 3 years longer in a kid with ADHD. So he'll grow out of it? Absolutely true for at least some of the kids. They just don't grow up until they're in their mid-20s. That's because these are cortical maps, and you can't see on these. These are essentially trajectories of volumetric MRI. So you could make a map, and it's actually been done looking at the connectivity between prefrontal cortex and the striatum. and you see this same actual lag. And it's not only a lag in the formation of the structure, it's a lag in the connectivity, so there's an appropriate crosstalk. And you can do it actually with resting state fMRI as well, looking at the default. There's all sorts of different things that do this. So back to this little diagram. So what is it about genetics that drives this process? Here we're now going to be looking at this level of analysis to see how susceptibility alleles, genetic variation, predisposes vulnerable persons to becoming mentally ill. So a gene is the fundamental building block of who we are. This is what evolved at the beginning of evolution, was the ability of deoxyribonucleotides to carry information. These are the sort of instruction tools for how you build an organism. And we've now been able to map from the Human Genome Project, map the human genome. And this was published in Science a couple of years ago, what's called the HAP map. So a gene, DNA is composed of these building blocks, AT pairs, adenine, thymine, and guanine cytosine pairs, each side of a double helix, like you can see this here. And there are about 3 billion of these things in your average human genome, comprised of 23 chromosomes. And the DNA itself, in order to take the information out of it and use it to build a cell, is transcribed. So you have these little gadgets called introns, which carry the instructions, for example, for making a protein. And this DNA is transcribed across these introns into RNA. And that RNA then runs through a ribosome and becomes a protein. And then those proteins do the things that essentially comprise the machinery of cells. Now, there are about 3 million polymorphic sites in human DNA, and a polymorphic site is one of these CG or AT pairs. So if you just run along with an A, you know there's a T on the other side of the DNA double helix. So if you just run along, for most people, most of the time, everything is identical. In fact, almost 99% of the time, it's identical with chimpanzees. About 50% of the time, it's identical with a fruit fly. So there's tremendous conservation of these DNA sequences. But now and again, you get DNA sequences that vary. You get one that's different. So you can see an A and a T here and a T there. And you can get repeats that are different. So this is a set of what are called repeats. Here's one version of this, and it comes along, and then here's another version and another version and another version. And sometimes things get deleted. So this is a deletion where you've got 2 bases that are deleted, and sometimes they get inverted, they get flipped around. And all this variation in genes is what accounts for evolution and what makes you and me different one from the next of us, because no human being, except one who was cloned, and probably not even then because there are mistakes made during replication, has exactly the same base pair sequence across all these chromosomes.
I have had cell and molecular biology classes, so this is I know what you're talking about here, but I'm just wondering if we could really take it and bring this back to how it ties into, you were mentioning alleles, and how this ties into mental health, and
so we'll get there.
And again, talking about what is gray matter versus white matter in a brain and the differences there, things of that nature.
So just be patient.
I will be.
So we now have, in addition to the human genome, we have genome maps for fruit flies, C. elegans, the worm, mice, fish, tigers, bears, all sorts of different plants. I mean, lots and lots and lots and lots of things have been mapped. And there are somewhere pushing 20 now humans who've had every single base mapped. The first 2 were Craig Ventnor and Jim Watson. And within probably 5 to 10 years, all of us, as a routine commercial thing, will be able to have our DNA mapped from the from the first base to the last base in our chromosomes, because the technology is getting so good. So with that information, knowing the pattern of variation in lots and lots and lots and lots and lots and lots and lots of humans, we can begin to say, how do humans differ one from the next? And we can do that in a way that allows us to ask that question relative to patients with an illness versus persons without that illness. So this is now using mapping the ability to map base to base your DNA and compare it with a person who has an illness, a patient group, and say something about variation, DNA variation that's associated with illness. This is called genome-wide association scanning, or GWAS. And Andrew Allen, for example, here is one of the world's experts in the statistical analysis of this in patients with neurodevelopmental disorders. So you can see here that this patient right here has a different pattern of alleles variation at these single base pairs than this patient over here. And it's actually, there's another one down here. There's an AG rather than an AT pair in this. And the ability to do this comparison then allows us to say, well, this person with this particular gene having this particular variation is vulnerable to mental illness, where somebody without that allelic variation is not vulnerable to mental illness, or prostate cancer, or you name it, whatever the disease state is. And there are lots and lots and lots and lots of genes that are now being identified as vulnerability factors for mental illness. And in fact, if you map the number of these GWAS analyses every week, it's not doubling, but every few months now it doubles. So it went 4 or 5 years ago from 1 or 2 of these things to hundreds and hundreds and hundreds, and more and more are getting published every day. So these are, sorry. Can you? Okay. So these are just some of the genes that have been identified based on allelic variation as risk factors for mental illness. And these are some that have been identified specifically for schizophrenia. And the one that I mentioned before, the one that's expressed only during fetal development, is this isoform of neuroregulin. And this is what happens if you have this vulnerability. This is what happens to your brain. So if you watch this here, this is excessive pruning. Remember I talked about apoptosis and cell death and rearrangement? Well, your brain basically goes haywire during your teenage years if you have schizophrenia, mostly driven by genetic vulnerability. This is like having a left main coronary artery heart attack in this patient's brain. It basically is disintegrating before your eyes, largely because of this genetic risk, coupled to things like prematurity, having maternal exposure to influenza in the second trimester, a whole variety of environmental risk factors. Okay. So genetic vulnerability now yields to, what does this do with respect to the process of assembling cells, and how does this express itself in mental illness? So here's a cell. This is an axon. When these things are built, because they carry electrical signals, they require shielding or insulation. So this is myelin around this axon, and what you see here is the cell body here, and these little gadgets called dendrites. And then down at the end of this long signaling mechanism, so the electricity goes this way to this way, you have this axon out at the other end, which will hook up on another neuron with one of these little dendrite guys. So you can see here what this looks like on an electron micrograph. Here's the presynaptic neuron, and all these little things in here, these little round guys, are called vesicles, and they contain the neurotransmitter, which when released across the synapse, this little gap here, triggers the postsynaptic neuron. These are dendritic spines, to fire off, and then the neurotransmitter is cleared out of this out of the synapse, so the neuron can reset itself. It's like an on-off switch. You've got to have some recycling and clearing mechanism, otherwise the switch would completely be on. And if you look back in here, these are very complicated structures, synapses. Here's that protein. Well, I guess neuroregulin's not on here, but there are about 1,800 known proteins in the postsynaptic density in a average human neuron. There are about 800 in a fly. So you can actually map the course of evolution as a function of the complexity of the postsynaptic neuronal density. So if you think about 1,800 proteins sitting in there and how you make sense out of the interactions between all these proteins across development, you get a sense for how challenging all of this is. And the reason it's challenging is because they do these things. They handle cell adhesion, how cells stay next to each other, get to where they're supposed to be, and then stay put. They handle neurotransmission. They handle, once the postsynaptic neuron has been stimulated, they handle what's called signal transduction, which is the process by which information is carried down the neuron and changes happen in the neuron, like neurogenesis. We'll talk about that in a moment. So these are very complicated cells, much more complicated. Any individual neuron is much more complicated than any structure at the cell level in the heart, for example. So here's an example, probably the best example, of a single gene neurodevelopmental disorder. This is a child with Fragile X syndrome. And you can see here he's a kind of happy, a little bit funny-looking kid. He's got kind of big ears and widely set eyes. And look how big his hands are. So there's a whole set of physical characteristics for kids who have Fragile X, which is the commonest known genetic cause of autism. These individuals are usually mentally retarded. Some of them have low average intelligence, but most are mentally retarded. They have a lot of cognitive and affective processing difficulties. Many of them have no language or limited use of language, and they are really sweet kids, most of them. So this disorder is a classic Mendelian X-linked recessive. It makes a protein called FMR1, this particular gene for Fragile X. And if you're missing this FMR1 protein and you have a copy, 2 copies, because your mom carries 2 X chromosomes, she gives one of those copies that's broken, where the FMR1 protein is broken, to a dad who doesn't contribute an X chromosome, and you get an FMR1 affected. This is an unaffected son, an unaffected daughter. Here's a daughter who's a carrier, and here's this little boy with Fragile X syndrome. So here's what happens with that FMR1 protein. This is where the transcription process starts, and you move down all these different introns that define the gene itself. You end up with a functional FMRP, and it is a transcription regulator. It essentially tells a particular signaling pathway not to be so active. It's kind of like a brake protein. And at the beginning of this thing in the DNA, there's this vulnerability to CGG repeats. And all of us, normal people, have between 7 and 52 of these CGC pairs. As these repeats amplify, when you get over 200, that blocks the transcription of this protein, and you get a broken FMR1 system. So that's how Fragile X happens. And you can model this process in the mouse. And if you look at a mouse who has this particular mutation, the neurons in the mouse, the Fragile X model mouse, look like this. So you can see they're skinny and misshapen. All those functions that we talked about a cell ought to be able to do don't work very well in this individuals who have this broken FMR system. If you use a drug that sits on the mGluR5 antagonist in the mGluR5 protein, the glutamate receptor, in the postsynaptic dendritic neuron, and you block this mGluR5 protein, you essentially mimic FMR1, the activity of FMR1, and you can return this broken system to this, and eventually over a matter of weeks to months, you reconstitute normal, the normal configuration of this cell-to-cell connection, this synaptic junction. And if you do that in mice, or you do it in flies, you completely reverse the behavioral phenotype of the FMR1 deficit. And we are now with Seaside Pharmaceuticals and working with Avi Lekevich, who runs the Fragile X clinic here, probably going to do the first in patients for Seaside's FMR1, Seaside's mGluR5 antagonist. And if it works, this will be the beginning of the first. I mean, it's an incredibly beautiful model system for what we're talking about in terms of the developmental neuroscience revolution. And it is an incredible privilege to be a part of that. And the people who've worked out the biology of this are distributed around the country, but many of them are at Davis. And I'm going to be headed out to Randy and Paul Hagerman's laboratory in a couple of weeks for their center grant in Fragile X. And we will, if it works out, in neurosciences medicine, be developing the clinical trial endpoint for, if it's successful, this mGluR5 antagonist to move it through the regulatory pipeline all the way into and through phase 3. So we'll see if we get lucky and all of that happens.
Dr. Marsh?
So they've done that so far in rat models. Have they done it in higher level animals? Because I'm assuming you're not going straight to kids.
It's been done in monkeys. Or actually, I should say it's being done in monkeys. And there are lots of toxicology studies with the drugs, some of which have been done in monkeys. There's a lot of interest in these compounds in autism because some of the same pathology is present in autism. The first in-patient study, which we had hoped to get here, is being done at Covance in Madison, Wisconsin. But I think we have a pretty good shot at getting the first in-patient single ascending dose study in the DCRU, which will lead to the multiple ascending dose studies, which will push us into phase 2.
What kind of kids age range are they doing? Little ones thinking they'll have a better impact?
It'll be done in adults first.
Okay.
And then it'll be brought.
Okay,
it'll
adult
be brought
Fragile
down.
X. Okay,
That's right.
got it.
It'll be brought down into children once it's been done in adults.
Thank you.
We'll talk about that later. It's an interesting question. So you have genes. Genes carry the instructions for building cells. Cells find out where they're supposed to go, and they begin to establish conversations with each other. And if that process, what's called cellular molecular neuroscience, breaks down, then you get disorders like Fragile X syndrome, or in some cases, disorders like OCD. There's a mouse model for OCD, the SAPAP3 mouse model for OCD that Guoping Feng here in the Department of Neurobiology developed, which is a broken scaffolding protein in that postsynaptic density. These cells then aggregate in groups, what are called hierarchically distributed neural networks. And you can think about them like a computer network, and their job is to process information. Cells process signals from one cell to the next, and then you aggregate across billions of cells, and you have the capacity to see things, hear things, do things, talk about things, think about things, feel things, and interactions between all of these various information processes that make us fully human. So here's an example of this. This is now moving back into the world of brain architecture. This is from Joe Ledoux's laboratory. And this is basically the circuit that's involved in processing fear. So if you see something that's scary, like a snake, it moves from the back of the retina and hits the visual thalamus, one of these relay nuclei. And before you have a chance to do anything, this is like blinking, right? You're already going to step back. There's no thought in here. It's like 10, 30 milliseconds later. Eventually, it goes back into the visual cortex, where you actually have an image, an upside-down image of a snake. That's passed to memory, so you know that this is a snake. You have a name for it, and you integrate that with all of your experience of snakes, whether it's a rat snake or a garter snake or a copperhead. That information is integrated with the fear response, right? This is an unpleasant thing for most people to see a snake. And all of that, what it looks like and what your reaction and what it is and your previous experience is then passed up into the prefrontal cortex where you continue to attend to it. This is this little gadget called the anterior cingulate that handles that attention step. And you make a judgment about whether you want to pick it up or you want to put it down, or God forbid you want to chop its head off, or whatever it is you want to do with this particular creature. And there are all sorts of autonomic responses, like your heart rate goes up and you get ready to move. And all these things are handled automatically without you having to do any. There's nobody up there running this. This all happens automatically. So if you look at this in terms of circuit models, here are genetic influences on this sphere circuit, which includes the amygdala, the prefrontal cortex, orbital frontal cortex, anterior cingulate striatum, and thalamus. These are the circuit stations or way stations that handle integrating all of this information. And when these things get busted, there's disruption in core psychological processes. In this case, for fear, the appraisal of threat and what's called habit learning. And when this gets bad enough, we label this as an anxiety disorder. So you can see how this works looking at this functional magnetic resonance image where Danny Pine at the NIMH intramural program took normal kids and kids with generalized anxiety disorder and showed them an angry face in the scanner and watch what happened to the amygdala, which is this emotion processing center that handles fear and anger responses. And you can see here that the amygdala stayed pretty quiet in the control group and went off in the patients with generalized anxiety. And if you look at it as a picture, right here, the left amygdala lit up in the patients as compared to the controls. Now, if you look at this schematically, this is what's actually happening. This is the anterior cingulate is right here, and this is medial prefrontal cortex. And this is the part of your brain that says, I know what this is, it's not dangerous, I can go forward. And every person in this room has had this. Anybody here who has never jumped off a diving board? Remember the first time it was scary? So the second time you got up there, it was still a little scary, but it was kind of fun. The fifth time, it was really fun. The 10th time, your mother was getting bored waving at you, right? And you wanted to go up from the low dive to the high dive. That process of getting over fear is called extinction. And the memory of having gotten over fear is called extinction retrieval. And basically what that means is that this part of the brain, the executive function centers in the brain, are able, in the sense of regulating feeling, to tell the amygdala to shut up. So you have this automatic fear response, and the amygdala says, oh, it's just a rat snake. Don't worry about it. Or, I jumped off the high dive last weekend. It was fun. I can do it again. Or, I can make friends. I can go and talk to that person, or whatever it might be. So there's a connection between this area, and that connection is busted in patients who have anxiety disorders, so that fear rules the roost instead of ordinary cognition. Now you can see this in post-traumatic stress disorder. PTSD is a disorder in which a person has a really horrible experience: rape, combat, a bad motor vehicle accident, really nasty, out of the ordinary things. And this is a schematic of all of the studies, fMRI studies, showing this failure of top-down control by the medial prefrontal cortex over the amygdala. And there are about 15 of them, all showing that this communication between the medial prefrontal cortex and the amygdala is broken. By the way, I could have shown you depression or other anxiety disorders. It's essentially the same story. So what's happening here is this top-down control step is busted. And it tends to be busted more in people who have a particular allelic variation in the serotonin transporter. So if you go back to the synapse, this is a presynaptic neuron, a serotonergic neuron. It dumps serotonin into the synapse. The serotonin diffuses across the synaptic junction and lands on the postsynaptic neuron and tells the postsynaptic neuron to fire off when that happens. To clear out this bunch of serotonin so that the switch can turn off, recycle itself, you can pull serotonin back into the presynaptic neuron. And that happens at this little gadget called the serotonin transporter, or which is the protein that handles the reuptake mechanism. And there's some other ways to get serotonin out of the synaptic cleft as well. But the primary way is through this reuptake mechanism. And the protein that does that for serotonin is the serotonin transporter. And there are two variations of this. One has a long allele in the promoter region of the transporter. This is long. And one has a short. And if you have this short allele, you are much more vulnerable to having depression and anxiety than if you have this long allele. And if you put patients in the scanner who have an anxiety disorder or depression, and you look to see what is the impact of the short form of the serotonin transporter versus the long form, what you see here is this communication step is busted in the short form of the transporter, so that the medial prefrontal cortex can't tell the amygdala to be quiet when it develops negative affectivity in response to some environmental stimulus. So we've gone from genes to cells to systems that process information. What about gene and cross-environment interactions? How am I doing time-wise?
Is it working? Yeah.
Okay. I have about 10 more minutes in the talk.
I had a question about the, you were talking about the serotonin uptake transporter there.
Uh-huh.
Does that presuppose that if someone, I guess, diagnosed with a depression disorder is, let's say, has the short version, they take said SSRI or whatever the case may be to offset the difference between long versus short, but in looking at it from this molecular perspective, are you suggesting that that's not going to change, or is there an evolutionary process of changing from the short to the long?
You're 7 slides behind, so we'll get to that.
Okay.
We'll actually get to an even more interesting variation of that question. So this is, you could get this idea that this is like a scientific story, right? It's intended to be like reading a novel. So the story is still unfolding. So one of the things that I said before, which is that you can't build a brain without experience. I mean, the brain is essentially a mechanism or a means by which we as individuals process experience, and that process of processing experience is how the brain is built as we age from birth to death. So you can't build a brain without experience, and these variations in the different risk genes that we are in the process of identifying themselves, the risk genes don't produce mental illness. It's the risk genes encountering some particular pattern of environmental experience. It's the interaction between those two things during development that produces these alterations in behavior, emotion, cognition, and perception that we label as mental illness. So this is a really nice study. Moffitt and Caspi published in Science in 2003. Very famous paper in the Annals of Gene-Environment Interactions. And what they did is they looked at a bunch of kids in Dunedin in New Zealand who they'd followed from babies on into young adulthood. And they had DNA on these subjects, and so they genotyped them for the long and the short and long version of the serotonin transporter, and they looked to see what happened to these patients' vulnerability to getting depressed as a function of which allele you had, long or short, and whether you'd experienced a traumatic event. So if you look at the people with the long genotype, these are the stress-resilient genotype folks, they didn't actually get depressed very much. And when they did, it didn't matter whether you had stressful events or not. It was all the same, from no stressful events up to 5 stressful events. But look here at the kids who had the short arm variant of the serotonin transporter. They got depressed much more. This is 35% of them who'd had 4 or more stressful events. Classic example of a dose-response curve for a gene by environment interaction. So if you have this short form, and you're a little like Eeyore in your temperament, and you have something bad happen to you, you're pretty likely to get depressed. If you have a really wonderful, nurturing environment and nothing bad happens to you, then you have no increased risk of getting depressed over folks who have the long form. But if something difficult happens in your life, this would be something like a divorce or a car accident or a move or worse, then you become more vulnerable to depression. Now, there's another way that genes interact with the environment called epigenetics. So if you go back and you remember what a chromosome looked like, it doesn't look like this double helix. It looks like all this clumped up crud here, right? The classic picture of a this could be, say, the X chromosome. So one way that genome regulates whether something is transcribed or not is to tightly wind the DNA together. And it does that through these proteins called histones, which sit kind of clumped along the individual bases of the double helix. And when these these stretches of the DNA are when these histones that promote this clumping, when they're acetylated, everything is tightly bound up, right? It's like the DNA is constipated. It can't do anything. And when they're deacetylated, then lo and behold, look what happens. The DNA begins to unwind, and when it unwinds, that allows for transcription to happen. There's another way that this can be blocked, which is to methylate the DNA at particular bases, which can also block transcription. So here are two ways that you can regulate your DNA that have everything to do with your experience and nothing to do with what the individual allelic variation is in your particular genome. Now, this is a mouse. This is from Michael Meany's lab up at McGill in Montreal. And this is a mouse, and the mouse has puppies, and the mother mouse is licking the puppies. So this is maternal licking and grooming behavior. And I want all of you to go home and lick your children after you hear this story, because this is a really nice and good and wonderful maternal licking mouse. And there are variations in this. There are mothers who do a lot of licking, and there are mothers who do very little licking. And everybody knows, you look in human populations, right? There's some nurturer mothers and fathers, and some non-nurturer, wire monkey mothers and fathers in the population. Well, it turns out, if you are a rat pup and you are exposed to a stressful event and you've had a nurturing mother, you're much more stress resilient than you are if you've had one of these mothers who doesn't do much maternal licking and grooming. And the interesting thing about this, I don't have it on this slide, is that this phenotype, this stress responsivity phenotype, is passed on in the next generation from mother to daughter. So you may, if you experience this as a mother, having a non-nurturing mother, and your DNA is structured for stress responsivity, even if you're a nurturing mother, you will pass this trait on to your daughter. So if you believe this, this is why programs like Head Start and early child intervention, the kind of things that one of the presidential candidates at least is talking about, are absolutely critical. So you can regulate your DNA through experience, which means that we need to pay very close attention to what happens in the environment. Now, that takes us to what does this mean for us folks who live in a clinical neuroscience world. All sorts of interesting things are going to be coming down the pike from the bench, and they're going to move in research settings, the DCRU, for example, the T1 translational research block, to the bedside in academic health centers, and out of academic health centers, hopefully into practice settings. So here's how it works for treatment. You've got an immature brain. Through genetic environmental factors, you end up with basically not much proneness toward anxiety. Or you might, from genetic environmental factors, end up with high anxiety. Now, if you are a high anxiety human being, you can get rapid therapy, like diazepine, which inhibits a neurotransmitter called gamma, which activates a neurotransmitter called GABA, which is the inhibitory fast-acting neurotransmitter in the brain, and you take a Valium and you calm down right away. Or you can do slow therapy, which is like psychotherapy or a serotonin reuptake inhibitor. And over 6 to 8 weeks, your brain will learn something new. You'll enable this medial prefrontal cortical control over the amygdala through a process called neurogenesis, and you will end up back in the low anxiety state. This is how that works. This is what's called neurogenesis. And it used to be thought in the adult brain that you didn't make any new neurons. That was the dogma until 5 to 7 years ago. Well, now we know actually all over the nervous system you can make new neurons in the adult organism. And in fact, the process by which you remember things, including remembering that it's actually safe to jump off the diving board, is a function of the ability to generate these new neurons. So if you don't make new neurons, you block this process of neurogenesis, then you block the ability of treatments like serotonin reuptake inhibitors or cognitive behavior therapy in mouse or rat models to work their magic. So that process of neurogenesis happens. It's driven by genes acting in the cells. So here's a serotonin, here's a serotonergic neuron landing on this glutamate neuron, which is the fast-acting activating neurotransmitter in the brain. And here's all the things that happen at a certain nergic synapse. And one of the things that happens is an activation of this thing called brain-derived neurotrophic factor, BDNF, which is one of the proteins, perhaps the main protein, which drives this process of neurogenesis. And if you have a particular allelic variation in BDNF, you'll see the same kind of picture that I showed you for the transporter for BDNF with respect to that prefrontal cortical amygdalar connectivity problem that you get with a short version of the serotonin transporter. So drugs like the serotonin reuptake inhibitors, these are a whole bunch of those little gadgets, act within the amygdala hippocampus prefrontal cortical circuit that defines mood regulation to restore top-down control by the medial prefrontal cortex over negative affectivity via neurogenesis, which is why it takes 6 weeks to happen rather than the couple of minutes it takes with a drug like Valium. Turns out the same thing happens with cognitive behavior therapy. So the way to think about psychosocial treatment going forward is to think about it exactly like we think about physical therapy. So here's somebody who's blown out a knee and has a physical therapist who's rehabbing the knee. The rehab process involves instruction, strength, flexibility, psychoeducation. Here's a kid in my clinic who has obsessive compulsive disorder and contamination fears. And I'm not rehabbing his knee, I'm rehabbing his cortical striatal thalamocortical circuitry, which is hiccuping in OCD by asking him to touch the toilet seat and not wash, all the while saying to himself that this is just OCD. I know what this is. You know, there's actually no reality to this fear. It's just my brain on hiccups. And if you do this over and over again, just like jumping off the diving board, it becomes fun instead of scary. Now, when I teach this technology, and I actually develop the protocols that are widely used now and considered the standard of care for OCD in kids, I'll ask how many of you would be willing to go in a bathroom with a kid in the building in which you work, touch the toilet seat, and then eat lunch without washing your hands. So raise your hand if you could do that. So I'm the only one in this room who actually could treat kids with OCD. And there are lots and lots and lots of people out there treating these kids. But if you can't go there, how do you expect the kid to go there, right? So I remember once in a lecture that I gave, I actually bought a toilet seat at the hardware store, and I stuck it on the table on the way out the door, and I said, all right, every one of you touch that thing on the way out the door. And it was amazing to what how much resistance there was. Well, why does that happen? It happens because we have built into the central nervous system things like disgust, fear of snakes, fear of heights, fear of the dark, fear of strange things, strange-looking people, right? These are fears that have been selected in evolutionary terms, and in some patients they get out of hand. And the only way you can get them back under control again is to do the opposite of what the fear is telling you to do. And it's exactly like physical therapy, except in this case we're rehabilitating a dysfunctional information process that has the patient off their developmental trajectory. And to get them back on again, you've got to teach them, the same way you teach a dyslexic to read, to do it properly over and over and over again. So essentially drill and repetition. Now, exercise.
I'm not afraid of the Home Depot toilet seat. I'm afraid of the one in the building. Okay? I just want to make that clear.
So there are some toilet seats that you simply would not want to touch. And so exposure only works in the absence of real threat. You know, somebody who's a sexual assault survivor, you wouldn't ask them to walk down a street at 2 o'clock in the morning in a really bad neighborhood because it's not safe, right? But it is safe to do this. It is absolutely safe to do this. So, exercise, great treatment for depression. New story out from Jim Blumenthal's group here today about exercise, depression, and heart disease. This is the impact of exercise in a rat model on BDNF expressed in exactly these structures we've been talking about, hippocampus and amygdala. And you can see here in the non-exercised rat, there's not much new neurogenesis. In the exercised rat, there's a lot of neurogenesis, and you can see BDNF levels rising in correlation with this, which is why, I don't know about you, but when I get grumpy and irritable and all stressed out, what do? I go for a run, right? It's a very powerful treatment. So all of these treatments, cognitive behavior therapy, exercise, serotonin reuptake inhibitors, meditation, whatever it is that you're doing, if it works, it's working via this neurogenesis platform. So the paradigm is pretty clearly changing. This is very different than the paradigm we've used in caring for mentally ill patients, even as recently as as 10 years ago, patients coming to Duke had a pretty good shot of getting psychodynamic psychotherapy rather than one of these more science-based interventions. So it's a little like the Kodak film camera giving up to turning into the Kodak digital camera. We've got to decide what to keep of the old technology while rapidly transitioning to the new technology. We're moving out of these models of psychiatric illnesses as stories that relate to how you got along with your mother, acknowledging that early life experience has a major effect on whether you will become mentally ill if you carry genetic vulnerability factors. And these are now seen as trajectory-based neurodevelopmental disorders. So from a science point of view, what we'll be doing is moving, oh crap. I just did that to wake you up. Yeah, no, right, I'm tough to teach. One more. Well, that's okay. Right there's good. So right now we have episodic care. People come in, they have a complaint. We have a diagnostic system that basically describes their phenotype. We don't have any biomarkers or biosignatures, but we are beginning to develop through this basic understanding of the pathobiology of how these information processes are built and when they fall off their developmental trajectories, a better model for molecular diagnosis and risk assessment. And eventually, over the next 10 years, we're going to move from this episodic model to a more organized model where we understand your genetic vulnerabilities and your environmental experience and your health history, and we'll move more in that context toward preventive interventions, actually trying to head these things off at the past. And eventually, we'll be able to bring this down into personalized health care, which will be, these are the words of Elias Zerhouni, preventive, predictive, personalized, preemptive, and participatory. Individualized medicine. Now, this will happen because of the availability of internet informatics. These are all sorts of different ways that you can now access the internet to take databases and merge them to develop new ways of looking at information within the framework of evidence-based medicine. It will happen because every individual in this room within 10 to 15 years is going to have your genome sequence from base 1 to the end. This is Jim Watson, who, along with Francis Crick, discovered the structure of DNA, the double helix, and was one of the first to have his genome sequence. This is Craig Ventnor, who headed the Human Genome Project, and if you could read this, you can see he's got the genes for sleep advance syndrome, lactose intolerance, heart attack, substance abuse, more heart attacks, tobacco addiction, novelty seeking, right? You can look at all this stuff and say, this is terrible. But that's what, you look around this room, raise your hand, right, if you're an ADHD kid. But it becomes adaptive if you find the right environment. If you don't go in and make a mess out of your life, these kinds of traits actually in a population are quite useful. It will also happen because of advances in the electronic medical record, so that all of this information and all the information from all the other patients who look like you or don't look like you will be available in a single gigantic harmonized data set, which is the goal for the Murdoch Center, the Kannapolis Project, which is the heart of the Duke Translational Medicine Institute. So we'll be able to take patients in the Duke Health System and in Durham County and in Cabarrus County, and we'll have a map of their DNA and their environmental risk and protective factors and their health history. We'll be able to use these huge thousands and thousands and hundreds and thousands of patients to relate all these variables from genes to cells to systems to people in their context and understand from this information how to generate risk prediction equations. And as part of that process, we'll do like the Fragile X study. We'll take discoveries based on pathobiology and bring them into out of the laboratory and into humans. Here in the DCRU, They'll move into phase 2 using the Duke mechanisms for site-based research, the SBRs. And then out of the SBRs, we'll move into the DCRI, take them into humans in network-based studies, and then on into phase 4 using structures, network structures like CAPTAIN and Rena Delors' primary care research network, so we can understand how these things work in patients in the settings in which they, in fact, live. Now, what will this look like if you're a patient in 2020? You will log onto the internet, and you will immediately be in a system that knows your genome from base 1 to base to the N, and all of your health history, and all of your family members' health history, and all of the health histories of everybody else who lives on the planet. And you'll plug in whatever it is that you've got that's bothering you, and it will go from this from your plugging in your symptoms into this gigantic clinical biomedical data warehouse, and using a lot of very complicated data mining technologies, which have embedded in them these risk prediction equations, it will know what it is that you've got, and out of the computer will spit a set of instructions, like with cognitive behavior therapy, for what you need to do to participate in your own health care, and it will send you down to the pharmacy to pick up your prescription, and you will not need to see a doctor. So the idea that we need more doctors rather than less is temporary. In fact, this technology will be far more sophisticated at making diagnosis and deciding treatment than individual physicians who are basically creatures of habit, don't keep up, make lots of errors, don't like it when you question them. I mean, there's all sorts of things that if you were rational about it, would suggest that a computer, given sufficient information, would be a much better care provider than a human being. And in fact, there are studies now doing computer-based cognitive behavior therapy showing that a computer-based technology, either IVRS or web-based, patients do as well as if they have an evidence-based therapy from a human being as compared to a control condition, and they overwhelmingly prefer the computer-administered or IVS-administered treatment because it's convenient. You can do it at 11 o'clock at night after your kids go to bed, and nobody asks you when you're talking to an IVRS system how you got along with your mother when you were 3, and all the kind of other things that therapists typically tend to get lost in. So people basically want to feel better. That's why they come, and this will be the technology that allows that to happen. So these are the various funding sources that I've used to develop over my career this particular perspective. But it isn't about me, actually, or it isn't about us. It isn't about any of us. If there's any merit to all of this, it's the kids and the families that participate in research. So the whole edifice of medicine is based on a small number of people, like everybody in this room, working with a slightly larger number of patients and families. And if there's any merit or credit due, it's to the families that participate in research. So, time for questions. Peggy? Sorry, I went on longer than I thought I was going to go on, but.
So I have 2 questions. Is the brain physiology of phobias the same as anxiety?
Technically, yes and no. Phobias are hardwired, much more than say, something like social anxiety. And PTSD is completely a function of an environmental event. It's not hardwired. So if you're afraid of a spider, I'll give you an example of this. There's a guy named Steve Sonomi at Wisconsin about 20 years ago who took cage-reared monkeys that had never seen a snake. These are monkeys that for several generations have been raised in a monkey colony, a rhesus monkey colony, or macaque monkey colony. And with these monkeys, if you put a snake in the cage, the monkeys loved it because it was boring living in a cage, and it was something new to do, which was to play with this snake. So if you do that same thing with a wild-reared monkey, the monkeys go ballistic with fear, and they cower in the corner and make all sorts of vocalizations about being afraid of the snake. Now, the interesting experiment is if you take the cage-reared monkeys and you introduce a wild-reared monkey for a couple of days, and then you take the wild-reared monkey out and then you introduce the snake, the cage-reared monkeys that formerly had no fear went ballistic with fear. So somehow just the experience of being around that wild-reared monkey without a snake anywhere in sight allowed them to catch snake fear. Now, nobody understands actually how this happens. It's a really interesting question. But these are different, the specific phobias in that sense. In the other sense, they use basically the same fear circuitry that's used in any other anxiety disorder. It all runs through the amygdala, medial temporal lobe, prefrontal cortex. It's just more automatic, the simple phobias.
So just quickly, my second one is, if you are predisposed to having anxiety, genetically predisposed to having anxiety, but you don't have, which is not bloody likely, but you don't have any impinging environmental factors, then, you know, what's your outcome?
So your chances of developing an anxiety disorder are higher than if you don't have these genetic vulnerabilities, because I don't know anybody that doesn't have a stressful life. There are lots and lots and lots of negative life experiences in every human being's life. In some sense, the problem of being human is a problem of suffering. So we all suffer. Everybody has bad things happen. Everybody gets old and sick and dies. So,
Oh, uplifting.
so it's how you deal with it. It's how you deal with it that matters. And if you deal with it skillfully, if you're lucky and you've got a charmed life and you deal with the negative things that happen to you skillfully, then you know you can do fine. And there are many people who spontaneously discover jumping off a high dive or going up a ladder. They don't need a therapist to teach them how to do it or a drug to help them do it. They just discover, if I do the opposite of what my fears tell me to do, you know, I can beat the suckers. So that's the best of all possible outcomes. There are very, very, very few patients who actually aren't treatable. But there are some, and we don't understand that very well. That goes back to that personalized medicine bit. We can help, within 12 weeks, 60 to 70% of patients with depression or anxiety normalize, which is pretty good in my book. That's better than a lot of other areas of medicine. But we still have 30, 40% of people that we don't do very well with.
When can you actually have this kind of testing done?
So if you go to 123andMe,
Yes.
123andMe is started by the wife of one of the founders of Google. And also Rupert Murdoch, I think, has maybe it's his wife who started. Anyway, there are two big companies that are doing genetic testing commercially. One is Decode, and that you have to run through your physician, but they do actually a bit better job of the diseases they look at. And the other is 123andMe, which Decode is $1,000, and 123andMe is $300, and you don't need a doctor. So you get on their website, and you plug in your information, and they send you a little tube to spit into, and you send it back, and 3 weeks later you get the login information for every SNP in the HAP map. It's about 600,000 SNPs. And it'll tell you for particular diseases what your risk of having a disorder is over people who don't have that particular allelic variation. So for example, my grandfather died of prostate cancer in his 60s. And I've been thinking about doing it because you can explain about 50%, 40-50% of the risk for prostate cancer with 5 genes. You could do a little better with 9 genes. Decode does 9, 123andMe does 5. Decode is more expensive, and I have to go through my doctor to do it. So I've been just sort of playing with this in my head. I think it's premature for most people, because if you look at the actual prostate cancer is unusual, Alzheimer's is unusual. Most of the genetics added risk is very small. Like there are about 30 genes now that are related to height, and they explain about 2% of the variation in height. Most of it's explained by diet. Look at North Korea. North Koreans are about 6 inches shorter on average than South Koreans. 6 inches, 3 inches. I don't know. I learned that from Sarah Palin. True story. It's the only correct fact. Sorry, am I still live? So, but it's basically diet. Same thing's true for diabetes. There are a whole slew of genes now that have been identified as risk factors for diabetes, and they explain under 5% of the variance.
I think this would be really helpful for those who do not know who their biological parents are.
Yes. So I'll tell you one interesting fact, and then maybe we can keep going a little bit if you want. But these GWAS studies, right, where you compare patients and controls, sometimes these are done with what are called trios, so that you have a father and a mother and an offspring who's ill. And you're looking to see the transmission patterns between patients who have the disorder and their antecedents and patients who don't have the disorder and their antecedents. And one of the things that turns out, if you look at this, actually, because you've now got mothers and fathers in the mix, and the DNA on their children, is you can look at paternity, right? You can look to see whether a mother is the biological mother of that child or the father is the biological father of that child. And consistently across all of these studies, about 20% of kids who think that their two parents are their biological parents, about 20% of the time they're not. One parent could be the mother or the father, roughly equal, by the way. Mothers fool around as much as fathers. About 1 in 5 times, somebody was fooling around. And this is for reasons that isn't widely talked about. But it's there. It's a very interesting ethical question. Alice, please.